Hello everyone, welcome back. So in this video, we're going to talk about how the muscles contract. So uh, we can get the muscles to contract if you hit it with some type of electricity or but in terms of your nerves, I'll draw it for you. We have these multipolar neurons that's going to have multiple poles and then it's going to have an axon on the other end. So you got these neurons in your brain and your spinal cord. You have these neurons, and then you got this axon on the other end. This is called axon terminal. So what happens is that in either your brain or your spinal cord, you think about moving your muscle. When you think about you moving your muscle, it's gonna go ahead and send a signal down the axon. So it sends a signal down the axon. This is the axon here, and the signal travels down to the axon terminal, and that's what's being shown right here as well. These are the axon terminals, axons of motor neurons. So the signal travels down and then they reach the axon terminal. Then the axon terminal is what communicates with the muscle. So it communicates to the muscle, the place where the axon terminal is connected. We call that neuromuscular junction. So where nervous and muscular system interact. And it kind of makes sense why it's called that, because the name neuron means neuron, and then muscular refers to the muscle it connects to. So the junction is where it's connected. So the connection between the neuron and the muscle and is where the neuron is going to communicate to the muscle. And if you don't have neurons connected to the muscle, then muscles cannot contract and the muscles will be useless. And a lot of times people who have muscular diseases like MS or ALS, there's nothing wrong with their muscles. Their muscles are just fine. People with ALS, it's their nervous system that's not really functioning. So since they're not sending correct signals to the muscles become weak and they become kind of shrunken, and we call that atrophy. So your muscle becomes and the muscles become atrophied, and um, that's what happens when the nervous system is not functioning properly. So let's go ahead and zoom in on the part of this neuromuscular junction and what that looks like. So this is a neuromuscular junction signal from the nervous system's signals muscle cell to contract, where this interaction happens. I'm not sure why this is called LM, which stands for light microscope. This looks like it's an electron microscope here. So it's a lot bigger. It looks a lot more clear and bigger than it would look with the light microscopes. This is scanning electron micrograph, which is using electrons to bombard and create the image. And computer is able to create the three-dimensional image. So this is what it looks like in a three-dimensional image. And this is what it looks like in a two-dimensional image. It's false colored. Actual neuron is not going to have these colors. So that's the neuromuscular junction here. That's what we're looking at. And you'll see in a second, when you get really close to the muscle, is that from here, it looks like that neuromuscular junction or the neuron is connected to the muscle here. But when we got really, really close, you're going to see there's a little gap there. Okay, so there's a little bit of electricity that's going to designate the information that's coming down from the neuron, and it's going to go to the neuromuscular junction here. And that's what happens, that it's not really connected. So there's this little gap. So this is the axon terminal, and there's this little gap between, and that's the muscle. So the axon terminal, you got these chemicals then get released, and they're going to bind on the receptors on the muscle, and they're going to basically activate the muscle or tell the muscle to contract. It's not as simple as this, but this is the first step. First step is getting neurons to talk to your muscles. And then the neuron that's being released is called acetylcholine. And this is acetylcholine receptor. It's receiving that acetylcholine here, and it's activating the muscles. Okay, so when you zoom in, here's the entire neuron. This is the entire neuron here, and that's the axon, and this is the axon terminal. They're basically zooming in on the axon terminal, and here's even zoomed in more. So let's look at that. So that's your neuromuscular junction here. And if you zoom in on it, you see that inside it, you got these little bubbles, and these are called vesicles. And the vesicles are made up of a membrane inside. So the vesicle, you're gonna have these chemicals on the inside, and in the vesicle, these chemicals are going to move towards the membrane. And when the vesicle basically eventually merges with the membrane, the chemicals get released. So what I'm saying is that you got this vesicle, and they have these chemicals inside. And then when this vesicle is going to get, let's say this is the membrane, and when it gets merged with the membrane, that's the vesicle again, these chemicals are going to get released, basically. So they get released. So I'm trying to draw a vesicle here. So these chemicals are going to start coming out, and then they are going to cross the gap, and that was the gap that was being shown on the last picture a little bit. So they cross the gap here. They merge with the membrane, and they get released, and they cross the gap. So let's go back. So they cross this gap, and then they bind with the receptor on the other side. And the receptors on the other side cause the muscles to contract. And that's acetylcholine. So the acetylcholine is a neurotransmitter. Neurotransmitter means it comes with a neuron and transmits across this gap. And so it goes ahead and it signals the receptors. And if there are receptors on the other side, they're going to cause a whole cascade of events that causes the muscle to contract. And this is one of those places where pharmacology can come into play. So if you're thinking about becoming a pharm tech or going to pharmacy school, um, so you could potentially get chemicals to come in here, and depending on the type of chemical it is, um, you can either, let's say, take a muscle relaxer that deactivate these receptors, even though acetylcholine is coming here. Your nerves, they are sending transmitters, or the receptors are offline. Basically, they're not receiving any signal, so they can't cause the muscles to contract. That's what muscle relaxers do. So something like anesthesia, you want to maybe make the muscles paralyzed where they can't move. That's what you can do with this knowledge, what to do when you want to maybe paralyze the muscles. So you can figure out how to use that knowledge in medicine. So we're going to continue with this, but let's talk about first how muscle cells grow. So we have that next. So you're developing as a fetus, we grow muscle, we make muscle first, and the thing that happens is that you got these little kinds of cells called myoblast. And if you remember from before, myo stands for muscle, and blast means to build. So these are myoblast cells. And then if you remember not blast, sorry, clast, that meant to destroy or break down. But anyway, so blast means to build. So you got these stem cells, they are going to develop into a muscle fiber, or skeletal muscle fiber in this case. So what they'll do is they'll start like you can think of Play-Doh. They'll start to form these big clumps, and they form these big clumps. And what's interesting is once they form these big clumps, the membrane kind of dissolves. And when the membrane dissolves, you first start off with myoblast, and you see that they have these nucleus on the inside, and once they kind of combine together, you have this bigger muscle cell, but you still have 4 nuclei here. So 1, 2, 3, 4. So you have 4 separate cells. They combined, merged together, and you can see that they have these nucleus or nuclei when you merge them together. 4 nuclei. That means when cells merge, they do not lose the nuclei. and you end up having more than one nucleus in each of these muscle cells. So if you logically bring it into conclusion, this is how you get really long fibers with lots of nuclei. So this is a long cell, so one cell, but it took a lot of those small little cells to create this one long cell. So as long as your muscle, we're talking about maybe your hand, you have these lumbricals. Those are, not very long. But if you look at your thigh, you got your quads, your rectus femoris muscle. Those are pretty long. So those muscles would have to, a lot of these cells have to come together to create that muscle. And then you have this muscle in your spine, erector spinae, its entire length of your back. And so it's a really long muscle. So each of these muscle cells, there's going to be, long, then it's going to have multiple nuclei because a bunch of these myoblast cells had to come together to create that long muscle. Okay, so there's more than one nucleus per cell, and that's basically modification of a basic cell where you learned about, in chapter 2 about generic animal cell that had a nucleus, that had a mitochondria, had ER, that kind of stuff. So that was a generic cell, and then you merge it to modify it, and this is how you turn that into a muscle cell. So what we're going to do right now is we're going to look at unique terminology that we use for muscle after it has been modified, and after you modify the basic myoblast cells into a bigger muscle cell. Very large with cells with multiple nuclei. So this doesn't look like anything like a regular animal cell. It looks really weird, but we start off with a regular animal cell, then it turns into this really long thing, and that's very specialized in function. So you got something called sarcolemma and sarcoplasm, sarcoplasmic reticulum, something called terminal cisternae, transverse tubules, myofibrils. So you got these different specialized structures. And just to remind you again, myo stands for muscle, and I think that's Latin. But then we have a word here called sarco, and I think that's Greek. So Greeks, they don't have a word for muscle. They basically call it flesh, which means sarco. So it means same thing as a muscle. So that's where the terminology comes from. They don't have a specific word for muscle, they just call it flesh, and because of that, we're using the terminology, and one of these terms are going to be sarco. So we're going to call it myo, and for example, myofibrils, and then in some places will be called sarcolemma, sarcoplasm, sarcoplasmic reticulum. So let's talk about why this is called the sarcolemma. So if you remember, we have plasma membrane. If you draw it a different way, so it's the same thing, but if you draw a different way, it looks like this. So we have something called the cell membrane. So sarcolemma means same thing as a cell membrane. So you got your sarcolemma, it's called sarcoplasm, cytoplasm, and then also your sarcoplasmic reticulum means endoplasmic reticulum. So this is kind of showing you what it is equivalent to in a basic cell. And then so terminal cistern is also part of endoplasmic reticulum. So I just want you to look at the muscle model that we have in the lab and be able to label all these different structures. So when you go to the lab, grab yourself a muscle model and then maybe label the structures. So anyway, so you have your basic cell, and then it gets modified and it becomes this big muscle cell. Once the modification happens is that cell membrane turns into something called sarcolemma. And why it's called sarcolemma is that if you remember, I told you that cell membrane has multiple names. It's called plasma membrane, it's called cell membrane, and it's also called maybe you remember this, plasma lemma. So that's gets modified into that's a historical name for cell membrane, which gets modified into being called sarcolemma. So that means that it's going to be cell membrane around the muscle cell. We have the cytoplasm in the regular cell, and you can put sarco in front of it. It's called sarcoplasm. And then you have endoplasmic reticulum, or ER, and then that you stick sarco in front of that is called sarcoplasmic reticulum. And then in the regular cell, we do not have terminal cisternae, so it's just part of the ER. In the regular cell, we also don't have terminal tubules or transverse tubules. So this is unique in the muscle or to the muscle. And then inside the muscle cell, you're going to have myofibrils, which are equivalent to microfibrils. Microfilaments, sorry. Microfilaments are going to be the protein. So microfilaments are going to be protein fibers. And, this is where you might be asking, if you lift really heavy weights and you get big muscles, does that mean I'm getting more muscle cells? The answer is no. You have the same number of cells. What's happening is your body's putting more muscle fibers in these putting more muscle fibers in there. So that means the same number of cells remains, but with the same number of nuclei, but it's adding more myofibrils in there. So one cell starts off really long and really thin, and now that cell is getting thicker and thicker because you're adding more myofibrils, and that's the strength. So what's happening is when you lift weights or when you go exercise, your muscles get bigger, and that's because of the number of myofibrils increasing. So inside the muscle here, you have these myofibrils, multiple fibers in each muscle cell. So there's one, there's another one. You can kind of zoom in on it and see. There are a bunch of them inside. So it's a myofibril, and you have a few here. This is also labeled here, myofibril. So you have a bunch of myofibrils or multiple fibers in each muscle cell. And we're going to have this thing called microfilaments. Microfilaments are made up of two kinds. You have thin filament and you have a thick filament, and they basically overlap. And that's what allows the contraction to happen. So let's look at that. Okay, so we're going to go and zoom in on part of the muscle. So we're zooming in on the myofibril here. So you have this zigzag type of line going on here, and this is called Z line. So that's the Z line. The reason it's called Z line is because zigzagging between, and then we call that this part one Z line to the next Z line, we call that part sarcomere. So that's sarcomere. Sarcomere gives skeletal muscle striations, basically. So we're going to talk about that as well. So the sarcomere basically is the smallest unit that you can still contract, unit of contraction. And it's made up of Z lines, thin filaments, and thick filaments. So it's a smaller unit that can still contract. So you can go ahead and take the myofibril apart, but as long as you have all these parts of one sarcomere, contraction still happens. So if you break it down, you take away some of these components, it's not going to work anymore, but you have to have at least one sarcomere for this to work. And you'll notice that some of the fibers are overlapping here. And there are light spaces and dark spaces in this. So pretend maybe your fingers, they're fibers, and where they overlap, you'll see it'll create dark and light lines. I wish I could show you guys, but I don't have my video on right now. But anyway, so there's it's kind of light here, and then it's dark. It's light again, then it's dark. That's what's creating these striations. And if you remember, we talked about the skeletal muscle having striations before. So these spaces make it light in color, and dark lines and light lines create these striations. So it goes from light to dark and then so forth. So let me see if there's another picture. This is a different picture, but it's basically showing light, dark, light, and dark, and light, and that's what's creating the striations. So anyways, going back here, you've got overlapping going on, and sometimes you don't have overlapping going on. And Z lines are basically the anchor point. So Z lines, the anchor, and this is going to be the anchor point for the filament. So let me see. Z lines are anchor points for the filaments. So these guys are anchored onto the Z lines, creating one sarcomere. So that's what holds them in place. And you have another one called thin filaments here. Thin filaments are the actin filaments. So it's made up of a protein called actin, and that makes the blue. The blue one here is the actin. And then you have thick filaments, the red ones here. So they're labeled. The thick filaments are made up of myosin. That's another protein. It's a little bit thicker. That's why it's called thick filaments. They are designated as the red ones. And it looks like they're just kind of free floating, but they're still connected to Z lines. And I'll show you another picture. They're connected to Z lines with something called titin, which is another protein. Okay. So the interaction between thin and thick filaments is described by sliding filament theory, and this is representing one sarcomere here. So this is what it's going to look like under an electron tunneling microscope. So basically, you could you could interlock your fingers, and you could you can kind of move them in a way that this picture is moving. So the thick filaments, the ones in the green in the middle, those are the myosin, and then the actin filaments are the red ones here. Thin filaments, and they're red, so they slide past each other, and that causes contraction. And when they go back, that's a relaxation. So contraction, relaxation, contraction, relaxation. So if you're looking at that picture, that's what's happening. So I showed you where all the structures are and what they're called, but I haven't really told you how it works. So it's not magic, it's not actually a bunch of it's actually a bunch of chemicals working together for this reaction to occur. This is a picture under electron microscope here. It's called TEM, tunneling electron microscope. And this is magnifying it 64,000 times. So it's the benefit of using electrons is going to magnify things, 64,000 times versus in the lab, we have light microscopes. You can only magnify things 400 times. So imagine, you're zooming in so much that you can clearly see a sarcomere here. So Z line, there's a Z line right here. There's another Z line right here. So Z line to Z line is a sarcomere. So you're basically zooming in on a sarcomere. And then I mentioned that thick filaments are connected to Z lines. And this is kind of showing how they're connected. So you can see they're connected to Z lines. And this is called titin. That's another protein here. So these thick filaments, they are connected to the Z lines like this, and this protein is called titin. So that's what you can see right here as well. So that's a little protein that's going to connect thick filaments to Z line. You can see in this diagram from the previous slide that it's connecting to the Z line. And again, you can zoom in on this more if you're having a little bit hard time seeing it. So that's what holds that in place. Okay, so the sarcomere again is the functional unit. It's made up of two filaments. One is thick filament and the other one is thin filament. Thick filaments are the myosin, and thin filaments are the actin, and they're anchored to the Z line. So the thick filaments are anchored to Z line with titin, and this is kind of showing how it works, but it's also missing some crucial stuff that we haven't gone through yet. So let's look at that. So they are the attachment points for sarcomeres. Okay, so this is breaking it down more. So you zoom in on the thin filament and thick filament, it actually looks like there's more complicated than what it looks like there. So these are your thin filaments. You take that, you zoom in on it, and it's gonna look like these little red balls. Those are actin. And these red balls are covered with something that looks like barbed wire, and that's called troponin. And where they basically interact, this is called tropomyosin complex or troponin complex. What this barbed wire is doing is covering the attachment points of actin. So the actin filaments, you have these balls and the individual actin molecules, but it almost looks like DNA if you remember. It looks like string of pearls, and you have these string of pearls, and you twist them, and then you twist them, and then on top of it, you're going to have this troponin or tropomyosin. It looks like a barbed wire, and that's going to be on the outside of actin filaments. It's basically going to wrap around the actin filaments. And then you have yourself these thick filaments, and you zoom in on one thick filament, looks like a golf club. So if you were to tease it apart, you got this thing, the golf club, and this is the head of the golf club, and this is the handle of the golf club. So you can imagine if you were to grab a whole bunch of golf clubs and you put them together, this is what it would look like. So those are the thick filament. And then I mentioned the titin would hold on to the Z line. That's the titin, and it would hold on to the Z line. So this is anchored in place. So this is what it looks like at a molecular level. So when it's functioning, it's going to look like this. When the muscle contracts, you'll notice that the line gets much darker from there. And then when it's relaxing, it gets lighter. And that's basically the striations. That's what's making these striations. They'll look either, thick, or it look thinner, depending on if you're looking at muscle that's been contracted, or if you're looking at muscle that's been relaxed. So this is under the scanning electron microscope again. And this is the animation is kind of showing you the thick filament and the thin filament that they overlap. And in reality, this is what the micrograph looks like. So you see this, and this is what's causing the striations. So now we got all the components that we need to know. So let's go ahead and actually make the muscles contract. So what I didn't mention was that back to when I was talking about the neurotransmitter, that when the acetylcholine gets released and crosses the gap, they bind to receptors. So when they bind to receptors, it causes muscle to release calcium. That's what's going on here. So calcium is getting released, and the first thing that happens is muscle releases calcium, and then nerves goes to the, so your nerves send the signal, muscle releases calcium, and then the neurotransmitter stimulates the muscle, and the calcium that gets released, it comes from the sarcoplasmic reticulum. That's what it is here, sarcoplasmic reticulum, and that's equivalent to endoplasmic reticulum in a basic cell. It's going to store calcium. So you got this signal, it comes down, it tells the muscle, hey, release the calcium. And when the calcium gets released, the calcium goes down, and it binds to the troponin complex, the barbed wire that we saw on the last slide. What that does, it causes the troponin to move away from the actin filaments, and it exposes the binding site for the myosin head to bind. So calcium comes down, changes the position of the troponin complex. Myosin head can grab onto it, and it changes shape like this. And all of this is happening because you have ATP. So this doesn't happen magically. You need to have energy for it to happen. So the energy comes from ATP. And when the energy gets released, this ATP is going to become ADP. D stands for di, so two phosphates. So this is what it looks like basically. So you have this actin, and these black-eyed peas are the little circles that the myosin binding site is usually covered with the troponin complex. But once the calcium gets released and binds to the troponin, it moves the complex out of the place and exposes the heads for the myosin to bind. So the myosin binds there, but in this whole thing, the ATP is very important. So the myosin binding site is where myosin head binds. So you know what it looks like. Myosin can bind to it. Then you'll notice from the previous images that troponin, that little barbed wire is actually covering these heads. So when you don't have calcium, these are covered. You can't have binding. When the calcium comes in, it grabs onto the troponin. Troponin rolls over, exposes the binding sites, and then a myosin head can bind to it. So that's the function of calcium. It comes over, it binds to the troponin complex, and then here it's a zoomed in version. So here, this wire thing is covering the dark spots. That's where the myosin heads bind. And once the calcium comes in, it binds to the troponin and troponin complex, and then moves that wire away. Now the heads are exposed, and now the myosin heads can bind to it. And then it shifts, and it will cause the one filament to slide past the other filament, and that's how you have contraction. Here's another one. So you see the ATP is going over there, and the myosin grabs onto the actin and causes it to shift. And when it shifts, you have a contraction. And you can see that the little black, you can't actually see them here, they're covered. So, but anyway, so they bind, and that's what makes the myosin head move the actin. So when I learned this in college, it went right over my head. So if it's happening to you as well, right, just write it down step by step, and it's gonna start making sense. Oh, here you can kind of see that sliding past each other. So the in this case, the black bars represent the myosin heads. Okay, so the first thing that happens, so this is written down for you step by step. So calcium is released from the sarcoplasmic reticulum. So obviously the nerves cause it to do that. The nerves come and they cause the calcium to be released. The calcium binds to the troponin. And the thing that looked like barbed wire, that was troponin. And the troponin complex is going to shift orientation. It's going to expose the myosin binding sites. Myosin binding site is exposed, and then they are the myosin comes down, forms, it cross bridges with the actin. And one thing that is necessary here that you have to make sure that you have is ATP. So with ATP. And then actin filaments, they get pulled towards the center of the sarcomere. So cross bridges. And it connects like that, forms a cross bridge, and then ATP causes it to change shape. So ATP also causes the change shape. And then the two filaments, they slide past each other, and that causes the sarcomere to shorten. And this whole thing is basically called the sliding filament theory. So it took a long time for researchers to figure out what this was, and the working name for it was the sliding filament theory. But now we have it pretty much figured out, and we know how muscles contract. Okay, so here is another summary of it. You got yourself actin, which is in this case is showing yellowish spots, which is being covered with the troponin. And then once the heads get exposed, the myosin binds to it and makes it move. But in this whole thing, ATP is very important. ATP causes it to latch. ATP causes it to change shape. ATP causes it to release. So ATP is really, really, really important. It's required for contraction and it's required for relaxation. This is figure 9.7, and it breaks it down even further. And then we have another figure 9.9 here. And it's another diagram that just breaks it down even further. So you have all kinds of study resources available to you for help and you to understand the sliding filament theory. If my verbal explanation doesn't make sense, you can even Google. You can also go on YouTube and watch videos. So I just Googled sliding filament theory GIF here, and a bunch of stuff comes up. So you can use different resources, whatever makes sense to you. You can go to YouTube. You can even watch Khan Academy. This is very universal idea that's necessary for science students to understand how your muscles contract. So all kinds of different places where you can get your information on this. You can use your textbook. You can use your videos, and you can watch YouTube. So now this is how your skeletal muscles and your cardiac muscles contract, right? So remember, only skeletal muscles and your cardiac muscles has striations. It has these alternating dark and light bands, and that's found in these muscles. But what about the smooth muscle? Smooth muscle is not striated, if you remember. So how does the smooth muscle contract? So it does it differently, and that's what we're going to talk about in the next video.